Okay, welcome back, everyone. Let's get through the biology section now. So, as we know, we have reviewed the chapters 5 to. We have gone over the chapters 5 to. So, we have here, as you can see, the cardiovascular, just 7, immune system 8, digestive system 9, the respiratory system, which is 6, and the kidneys and skin, that is chapter 10. Now, there are flashcards to review for these, and we need to do the overviews, but I think I'm gonna pass on the overviews and we can go with the next chapter in biology. There's also biochemistry that we need to do with this one. So let's see. I don't mind honestly starting from chapter 1, 2, but here in our Jack Weston practice questions, we do have discrete questions. Unseen. We have 9 content areas, so there are separate questions on the nervous system, which I think is before the respiratory. So yeah, we have the nervous system here. We do have musculoskeletal, so we can do any of them here. And we have the viruses in prokaryotic life, which is chapter 1, I believe. Eukaryotic cells and membranes, that must be chapter 1, and that's chapter 2. And then DNA and genetics is the last chapter, and DNA should be on the 3rd chapter or so. So we can also start with eukaryotic cells, the first chapter here, the cell. Yep. So I did some reviews here on the first section, so I know now that the nucleoid region is a DNA region in prokaryotes. So we have the nucleoid, which is for prokaryotes, and we have the nucleoloid, which is for I believe eukaryotes. We have the nucleolus, which is it sits in nucleus and it has no membrane. So in eukaryotes, there's ETC in the mitochondria, the electron transport chain that happens there. But in prokaryotes, it happens in the cell membrane. There are large ribosomes in eukaryotes, but in prokaryotes, it's small ribosomes. And eukaryotes reproduce via mitosis, but prokaryotes reproduce via binary fission. And for prokaryotes, plasmids carry DNA material. So the nucleoid region should be uh, should be related with plasmids, and plasmids are in prokaryotes, and they carry DNA not necessary for survival. So the DNA necessary for survival is probably in the nucleoid region. We need to have Chat GPT here. I had here, so let's continue from here. So it's about plasmids and nucleoids. So DNA is found in nucleoid region. The DNA in the nucleoid isn't enclosed within a membrane-bound nucleus, which is a key distinction between prokaryotes and eukaryotes. The nucleoid contains the bacterial chromosome and associated proteins. These are small circular The plasmids are small circular extrachromosomal DNA molecules found in some bacteria and archaea. Plasmids exist separately from the bacterial chromosome, and they can replicate independently. They often carry genes that are advantageous to the bacterium under specific conditions, such as antibiotic resistance or the ability to metabolize certain substances. While both plasmids and the nucleoid contain genetic material, in prokaryotic cells, they differ in several aspects. There is structure. Plasmids are small, distinct circular pieces of DNA. Function. Horizontal gene transfer. Do we have it here? Horizontal gene transfer. I don't know why my alarm started now. Yeah, I think it was snoozed, that's why. Okay. I have my biology section here. Actually, let's do the cell. here. No, I mean B, B, C. Then we can do something here. The cell. So differences and similarities between plasmids and nucleoid. There's also something about nucleolus. So nucleolus is found in eukaryotic cells and is primarily involved in and it is a membrane bound nucleus. So the nucleus makes ribosomes, sits in nucleus, and it has no membrane. We have peroxisomes that collect and break down material. We have rough ER, which accepts mRNA to make proteins. We have smooth ER, which detoxes and makes lipids. We have the Golgi apparatus, which modifies and distributes proteins. So it packages, modifies, packages, and distributes proteins. And it is only in eukaryotes. Then we have the vesicular transport and cisternal maturation. So in cisternal maturation, vesicles travel in retrograde, new cis medial trans exit. And then we have COP2, which goes forward, and COP1, which goes backward. It is for return. We have peroxisomes, which collect and break down material. We have centrioles. These are 9 groups of microtubules, which pull chromosomes apart. We have lysosomes, demolition and recycling center made by Golgi, and it has single membrane. So the function, structure, and replication and inheritance. So these are bacilli, which is rod shaped. These are cocci, and these are spirilla. And we have obligate aerobes, obligate anaerobes, aerotolerant anaerobes, and facultative anaerobes. These are about bacteria. So obligate aerobes and anaerobes, it's explanatory, self-explanatory, since obligate means it is obligatory for aerobes to have oxygen and for anaerobes to survive without oxygen. And then we have aerotolerant anaerobes, so they can survive in oxygen, but they prefer no oxygen. So they do not use oxygen, but they tolerate it. And facultative anaerobes, they toggle between aerobic and anaerobic. There's also a fifth one which is not mentioned here. But for now. So we have gram positive is purple and thick peptidoglycan lipoteichoic acid wall. So peptidoglycan refers to the fact that it has both peptides and carbs. And the thin gram negative are pink, which the peptidoglycan cell wall is thin, and there's an outer membrane. We discussed the prokaryotes versus eukaryotes, and so what are the examples of prokaryotes? So bacteria, archaea, methanogens, halophiles, thermophiles, sulfolobus. So how about yeasts? Unicellular fungi that are classified as eukaryotes. They do have a membrane-bound nucleus and other organelles enclosed within their cells. They belong to the domain Eukaryota. So electron microscope images of yeasts. Cell membrane nucleus. What is the V? These are super cool. Look at them. Interesting. And then we have cyto. So for miscellaneous, we have prions and viroids. So viroids are plant pathogens, and prions are infectious proteins. They trigger misfolding, alpha helical to beta pleated sheets. They decrease solubility. So then we have cytoskeleton, which is microfilaments actin, and thick filaments microtubules tubulin, and intermediate filaments keratin, which equals vimentin, and desmin, which equals lamin. And then we have tissues, parenchyma, functional parts of organ. We have simple one layer, stratified multiple layers, pseudo stratified one layer, looks malt, but really just one, and looks multiple. Cuboidal, cube shape, columnar, squamous. We have gone through this for the skin part of biology. We have connective, which is stroma. They support extracellular matrix. Then bone cartilage, tendon, blood. This stroma is which support extracellular matrix. Yeah. Then we have genetic recombination as transformation, which gets genetic information from environment. And then we have conjugation, transfer of genetic info via conjugation bridge. Transduction and transposons. Transduction transfer using bacteriophage, and transposons genetic info can insert and remove itself. So we have different types of genetic information going in and out. Then we have viruses. They have capsids, the protein code. They have the envelope, which have lipid envelope. They have virion, individual virus particles, bacteriophage, bacteriovirus, tail sheath, injects DNA or RNA, and then viral genome. It may be a DNA or RNA, single or double stranded. And then if single stranded positive sense, it can be translated by host cells, and negative sense RNA replicates must synthesize a complementary strand, which can then be translated. And then we have retrovirus, which is single-stranded RNA, and reverse transcriptase needed to make DNA. So retrovirus is a single-stranded RNA. That was on my real MCAT exam. So if the passage says that this virus is a retrovirus, you know it's a single-stranded RNA, and you need reverse transcriptase, which is an enzyme, to make a DNA. Then we have bacteriophage life cycles, which is lytic and lysogenic. So the lytic life cycle is virions made until cell lyses, and lysogenic is virus integrates into genome as provirus or prophage, goes dormant until stress activates it. Is there anything else related to bacteria and viruses? Not. No. So there are specific steps and subcomponents for each of these genetic recombination methods and mechanisms. So we have transformation, transduction, and transposons. Transposons, they do genetic info. They can insert or remove themselves. They are genetic info. They can insert. So transposons can enter by themselves and exit by themselves. And we have conjugation, which is via a conjugation bridge. Okay, let's go for the first chapter. The cell. Perfect, we have it. It's 71 cards, so let's do this, and then this, and then this, and then this, and then this. Yep. Some of them should be duplicates, so that's why. Lysosomes are the what and what center. We know they are recycling center, but what else? Demolition center too, and they are made by Golgi. So Golgi makes lysosome. Golgi makes lysosome. Something toxic enters the cell lysosome. Who's there? I have enzymes and I'm not afraid to destroy myself. So they destroy themselves too. That's important. What contains 9 groups of microtubules? These are centrioles, and they pull chromosomes apart. So centrioles. Know that for cilia and flagella that the structure is a 9+2 arrangement made out of microtubules. So for cilia, which we had it in our digestive system, and even I think respiratory system, and flagella, which is so we can ask, what is flagella? Eukaryotes or prokaryotes? And I guess superloyal 9+3 pattern for centrioles. What the fuck? Is this a typo or are these two different things? Are centrioles different than centrioles. So we have daughter centriole, microtubule triplet, interconnecting fibers, proximal ends, distal ends, mother centriole, distal appendages, subdistal appendages. We got it, except then the 9+2 for flagella. A plasmid is a unnecessary DNA molecule separate from genetic DNA, chromosomal DNA. It's a small prokaryotic DNA. So plasmids are circular. Often the genes carried in plasmids provide bacteria with genetic advantages such as antibiotic resistance. So they are separate from the chromosomal DNA, but they, for example, contain antibiotic resistance genetics. They provide them with genetic advantages. What makes up part of the cell's cytoskeleton? Microtubules and microfilaments. Polymers of actin. We have microfilaments here. What helps a cell resist compression forces? So it would be cytoskeleton, microtubules. They help the cell resist compression forces. Made of tubulin proteins. These are thick filaments, 13 polymerized dimers of alpha and beta tubulin. Tubulin proteins, microtubules. And go down like this. Obligate aerobes require oxygen, O2. So micro aerophiles is the other one. So they're a bit less oxygen dependent than obligate aerobes. Obligate anaerobes die in oxygen. They literally die, so they go at the bottom of the tube. Facultative anaerobes toggle between aerobes and anaerobes. Anaerobic, so we have them dispersed all over. They're a bit more to the surface of the water than the aerotolerant anaerobe, since it will since they can toggle, but aerotolerant they don't require oxygen. So aerotolerant do not use oxygen but can survive it, tolerate it. Gram positive is purple and has a thick wall. Wall is made of peptidoglycan and lipoteichoic acid. Gram negative is pink and has a thin wall. It has an outer membrane too. Wall is made of peptidoglycan. What is when bacteria gets genetic info from the environment? It would be transformation. So when you get it from the environment, you transform. Griffith experiment with mice. R strain is safe and S strain is deadly. The bacterium, transferred gene, competent bacterium. They get it from the environment, from like a dead bacteria, and it's transferred. So transformation. S deadly, R is safe.
Welcome back. In this video, we'll be discussing the bacterial genetics, and we'll talk about transformation, transposition, transduction, and conjugation. We'll learn their meanings and how to differentiate between them. So first, let's talk about transposition. The bacteria has its DNA in two parts, either the bacterial chromosome or the bacterial plasmid. The bacterial chromosome is much larger and more complex and contains most of the bacterial DNA. And the bacterial plasmid is a small part of the bacterial DNA that has left the chromosome and now is floating inside the bacteria. So transposition refers to the process of extracting a small part of the bacterial chromosome to become plasmid or reintegrating the plasmid into the bacterial chromosome. Keep in mind that if the bacteria want to share its DNA with another bacteria, it can only share its plasmid, not the chromosomes. So the plasmid is smaller and readily accessible. Just remember that transposition has to do with plasmid. Now let's talk about conjugation. So some bacteria have a sex balance, and we call these bacteria F positive, and other bacteria don't have this sex balance, and we call this F negative. Keep in mind that the sex balance is encoded by the bacterial plasmid, not the bacterial chromosome. So the F positive bacteria will link with the F negative bacteria and form the mating bridge. Then the F positive bacteria will make a copy of its plasmid, which encodes for the bacterial sex balance, and will transfer this copy to the other bacteria. So now both bacteria have a plasmid which encodes for a sex balance, and both are called F positive. This is the process of conjugation. Transformation, which is also known as competence, is the ability of the bacteria to take naked genomes or naked DNA from the environment and use this DNA. So for example, if we have some medium with some bacteria in it and we put some raw naked DNA which encodes for antibiotic resistance into this medium as well, these bacteria will be able to take this DNA from the environment and use it, and it will become antibiotic resistance. However, if we add a DNA lysing agent into the environment, it will degrade the DNA and the bacteria will not be able to use it. We see this exclusively in Streptococcus pneumonia, H. influenza, and Neisseria. So remember, transformation for fresh DNA. And finally, transduction refers to the process where a bacteriophage, which is a bacterial virus carrying its own DNA, will inject this DNA into the bacteria. This viral DNA will be integrated into the bacterial DNA and will start replicating the virus. If this results in a bacterial death, we call this a lytic phage. And if it didn't, we call this a lysogenic phage. If the bacteriophage did not kill the bacteria, aka a lysogenic phage, it will give the bacteria some superpower. So the bacteria now can produce exotoxins. Alright guys, that's everything for today. I tried to explain everything as much as I could.
Subscribe. Oh my goodness. Okay, so transformation, fresh environmental DNA. So transformation, wait, not here, let's put it here. transformation, fresh environmental DNA. Let's put it here. Okay. Conjugation is a transfer of genetic info via a conjugation bridge. So we have recipient cell, donor cell. Is this to the same link?
Welcome back.
Yeah, so let's look at conjugation.
So some bacteria have a sex balance, and we call these bacteria F positive, and other bacteria don't have this sex balance, and we call this F negative.
Is this saying sex palettes? What is conjugation? And how does it work for bacterial genetic info transferring? What are sex palettes? Oh, we're talking about positive, minus, negative is like a sex, like couple. So we have genetic exchange, recipient cell, pilus, donor cell. So donor cell is donating these chromosomes, and there is a genetic. The conjugation bridge is called a pilus. What is the transfer of? I'm gonna put that as hard. What's the transfer of genetic material using a bacteriophage? So we have for bacteriophage is here. Wait. It's transduction.
And finally, transduction refers to the process where a bacteriophage, which is a bacterial virus carrying its own DNA, will inject this DNA into the bacteria. This viral DNA will be integrated into the bacterial DNA and will start replicating the virus. If this results in a bacterial death, we call this a lytic phage, and if it didn't, we call this a lysogenic phage. If the bacteriophage did not kill the bacteria, aka lysogenic phage, it will give the bacteria some superpowers. So the bacteria now can produce exotoxins.
So what doesn't kill you makes you stronger is definitely true for transduction, where the lytic phage is when the bacteria dies. Generalized its packaging, but specialized is excision. So lysogenic phage may result in bacterial death or give the ability to produce exotoxin.
Produce exotoxins.
Exotoxin or toxin, whatever. Let's make a screenshot of this bitch. Save to clipboard and put it right here. Cool. Okay. What else? What are DNA sequences that can change their position within the genome? within the genome, they can change their position. Transposons. This sometimes creates or reverses mutations. So transposons are DNA sequences. They can insert or remove themselves within the genome. These create or reverse mutations. So inverted repeats, genes for transposition, the inverted IS structural genes. Ask me GPT, ask me a challenging MCAT style question about transposons. Let's go. Okay. Prokaryotic cells reproduce via binary fission. It's a cell division in prokaryotic cells. Asexual reproduction, in which one cell divides into two identical daughter cells, is the equivalent of mitosis. Prokaryotic chromosome, plasma membrane, duplication of chromosome, division into two cells, division into two cells.
Let's talk a little bit about reproduction. Now, on Earth, and who knows, if we go to other planets, we might find new ways that organisms can reproduce. But on Earth, there's two primary ways that organisms reproduce. The first is that, let's say this is some type of unicellular organism. It essentially just makes a copy of itself. So that's it there, and then that's its copy. And in this situation, they have identical genetic material. So let's say this is genetic material of the first one, and this is the first one to begin with, and now you have the second one. Now, this type of reproduction, where one organism by itself can turn into two organisms, this is known as asexual reproduction. Or another way to interpret asexual is it is not sexual. So the next question is, what is sexual reproduction? This is where two organisms, or at least cells from two organisms, need to get together in order to produce a third organism. Here is a cell from one of the organisms. Let's say that cell is from the mother. It could be an egg cell. Here's a cell from the other organism. Let's call that organism the father. This would be a sperm cell. And when the egg and the sperm get together, when they fuse together, they have genetic information from both parents. And that new cell can become the offspring. And if sexual reproduction sounds familiar, it's because that's how you came to be. Now, to make this a little bit more tangible, let's look at some cases of asexual reproduction and sexual reproduction. So right over here, we see a diagram of a type of asexual reproduction known as binary fission. In general, if you hear the word fission, we're talking about splitting something. If people are talking about nuclear fission, they're talking about splitting of the atom. But binary fission, we're talking about one organism splitting into two organisms. And you can see that you have this genetic material, the genetic material, it replicates, and then the organism splits into two identical organisms, or at least identical from a genetic point of view. Now, binary fission applies to prokaryotes that we talk about in other videos. These are organisms that don't have organelles, that don't have a nuclear membrane, they don't tend to have multiple chromosomes. When we talk about eukaryotes, things that do have nuclear membranes, things that have multiple chromosomes, things that have membrane-bound organelles, you have a similar type of reproduction. This right over here, and we'll study it in depth in biology, this is mitosis. And mitosis, you can do this, the analog of binary fission, but we're now dealing with eukaryotic cells. Eukaryotic cells are more complex, so this process is more complex, but at a high level, they're both asexual reproduction. In both cases, one cell, or one organism in some case, has turned into two organisms that have the same genetic information. And it's worth calling out when you take one organism and you produce another organism with the same genetic information. It has a word associated with it, and that is a clone. And you might be familiar with the term clone from science fiction movies, and they usually use it in a somewhat related context. In Star Wars, when they talk about the Clone Wars, all of those clones, they all have the same genetic information as the original bounty hunter from which they were derived. Now, mitosis might also seem familiar to you because this is how most of your cells in your body divide. And this is how you grow. This is how you went from being a unicellular organism to being a multicellular organism. Now, you might be a little bit confused at this point. I just told you that most of your cells have experienced or are experiencing mitosis, but you're also the outcome of sexual reproduction. Well, what we'll see in a second is, as soon as sexual reproduction occurs, then this newly formed cell right over here, through mitosis, forms the multicellular organisms, the types that are you and me. But we also, as we grow and develop, we also produce what are known as gametes, that each have half of our genetic information. For human beings, this would be the egg produced by a female, and this would be the sperm produced by a male. And when they get together, the sperm fertilizes the egg. Well, then they can produce a viable cell that can turn into an offspring. Now, along the lines of binary fission, you have another type of asexual reproduction, in which case a mother cell buds off a smaller daughter cell. So like binary fission, it has to replicate all of its DNA, and then that DNA gets inserted into the daughter cell, but the end product, you have a bigger cell and a smaller cell. And we would say that the smaller cell has budded off of this larger one. And so this we call budding, another form of asexual reproduction. When we think about multicellular organisms, there are some typical types of asexual reproduction. For example, starfish are famous for their ability to form a new organism from a piece of an original organism. So let's say you were to cut that part off of the starfish. For many species of starfish, that could then grow into a new starfish, and the original starfish would grow that back. And so this type of reproduction is known as fragmentation. It's pretty easy to fragmentation. It's pretty easy to remember. You take a fragment of it, and it grows into another organism, and that's how it reproduces. Now, another fascinating type of reproduction is known as parthenogenesis. It's a fancy word. Parthenogenesis. And what happens here is you have an organism, and this is a baby Komodo dragon right over here, that can develop from an unfertilized egg. Remember, in sexual reproduction, we talk about how a sperm from a father organism needs to fertilize an egg from a mother organism, and then once they fuse together, then that fertilized egg can go through mitosis to produce a multicellular organism. But in parthenogenesis, you have an egg on its own that does not need to be fertilized, that can turn into a multicellular organism like a baby Komodo dragon. Now you might ask, well, how can this be? Well, sometimes these eggs don't have half the genetic material like we're used to with the eggs from sexual reproduction. For parthenogenesis, many times this egg has a complete set of genetic information, and then it can just go and start doing mitosis and turn to an organism. Or there's even situations where the egg does have half the genetic information. We'll talk about this in future videos. This would be a haploid cell. It has half the genetic information of its parent, but the haploid cell in certain cases can develop into a full organism. And there are organisms like Komodo dragons that can reproduce both with parthenogenesis, which would be asexual, and in certain cases with sexual reproduction. So I'll leave you there. Two big categories. I'll draw a dividing line here so we don't get confused. All the stuff that I drew on the left, to the left of this yellow line, these are forms of asexual reproduction, where for the most part, one organism can make a copy of itself that has the same genetic information. On the right, we have sexual reproduction, where in order to produce a new organism, an egg cell and a sperm cell need to get together. The sperm cell needs to fertilize the egg cell, and each of them would have a sample of half of the genetic information from their parents. Now, I'll leave you with the final question. We should always be asking this in biology, is why do we think sexual reproduction emerged? It actually emerged much later in our evolutionary history than asexual reproduction. Well, one argument for it is it produces much more variation in its offspring. And if you have more variation in your offspring, if you have more variation in a population, you have a higher likelihood that certain variations will be better suited for the environment that you are in. I'll let you think about that a little bit more.
That's interesting. So there's a theory that
better suited for the environment that more variation in your offspring. If you have more well, one argument for it is it produces much more variation in its offspring. Why do we think sexual reproduction emerged? It actually emerged much later in our evolutionary history than asexual.
So sexual reproduction emerged much later than
reproduction. Well, one argument for it is it produces much more variation in its offspring
than the asexual reproduction.
If you have more variation in your offspring, if you have more variation in a population, you have a higher likelihood that certain variations will be better suited for the environment that you are in. I'll let you.
So let's put this where let's put it here. Parthenogenesis. I never heard that. So we got this right. What are infectious proteins and can trigger misfolding? So infectious proteins that can trigger misfolding. Prions. Do we have it here? I mean, yes, it's infectious proteins. Viroids are plant pathogens. That I know. Prions, infectious proteins, trigger misfolding, alpha helical to beta pleated sheets. It decreases solubility. Causes Creutzfeldt-Jakob disease. So let's go check this out.
Let's talk about subviral particles. They're called subviral particles because they're smaller than viruses. And one thing to keep in mind is that viruses and subviral particles together are all categorized as non-living infectious agents. And this is a mouthful, but let's break it down again. The non-living part should be reviewed for you now because viruses, as we know, are acellular. They're only made of a protein coat or a capsid, which protects the genetic information, which is DNA or RNA.
It's a bit too fast for me.
And sometimes a lipid envelope, which I haven't drawn here because not all viruses have that envelope. And because they're acellular, they're so small that they don't have organelles. And that means they can't make their own energy, which means ATP.
So what is acellular again?
And this is a mouthful, but let's break it down again. The non-living part should be reviewed for you now because viruses, as we know, and one thing to keep in mind, particles. They're called. Let's talk about subviral particles. They're called subviral particles because they're smaller than viruses. And one thing to keep in mind is that viruses and subviral particles together are all categorized as non-living infectious agents. And this is a mouthful, but let's break it down again. The non-living part should be reviewed for you now because viruses, as we know, are acellular. They're only made of a protein coat or a capsid, which protects the genetic information, which is DNA or RNA. and sometimes a lipid envelope, which I haven't drawn here because not all viruses.
So we call viruses acellular because they basically don't have any cells. They're not a living cell. And they are a protein code, so a caspid, plus the DNA or RNA.
Have that envelope. And because they're acellular, they're so small that they don't have organelles. And that means they can't make their own energy, which means ATP. And they also can't divide or reproduce on their own. And in order to do these two things, it means that a virus needs a host. So we've re-summarized what we know already, and that's why it's called non-living. And because they need a host, they're infectious agents.
So because they don't have organelles, they can't make ATP, and they also can't reproduce. So they cannot reproduce. So they need a host, a living cell.
So you might be thinking, well, I know that about viruses. So what are subviral particles and how do they share those? So non-living. And because they need a host, they're infectious agents.
Because they need a host, they're called infectious agents.
So you might be thinking, well, I know that about viruses. So what are subviral particles and how do they share those same traits? Well, let's talk about them now. There are two types of subviral particles, viroids and prions. And viroids are smaller
subviral particles
than viruses because they're only viroids and prions. And viroids are smaller than viruses because they're only made of a single strand of circular RNA. Up until very recently, they were pretty much only found to infect plants, which I hope you can remember because I've written this in green and I've drawn this funky little bush thing on top of some grass. And today, viroids have been found in humans in the case of hepatitis D. So
they're not only plant pathogens. They're found in humans too in hepatitis D.
How do viroids make more of themselves if they only are made of circular RNA? Well, you can think of this in terms of Cs, because it's thought to be catalytic RNA. So C for catalytic and C for circular. And catalytic RNA means that it can make or break covalent bonds, another C. And because it can do that, it can self-cleave to create more viroids. Now, there's one thing to really pay attention to. You don't want to confuse viroids with virions, because virions are what we call whole viruses. That means the protein coat plus the RNA, or maybe an envelope. Because once a virus gets inside of a host and the protein coat falls off, it's only genetic material at that point, DNA or RNA. So that helps distinguish between a virus outside of a cell and a virus inside of a cell. So remember, viroids and virions are different.
Nice. So virions and viroids are different. Since viroids are plant pathogens also found in humans for hepatitis D, they are circular RNA and they are catalytic. So they self-cleave themselves to create more viroids. That's very interesting. And we should not confuse them with virions, which is basically a whole packaged virus.
And now let's talk about prions, which are kind of funky. They actually were very recently discovered because scientists have always argued about whether or not proteins can be infectious by themselves. Prions come from the word proteinaceous infectious particles. And
so prions have been discovered more recently, and scientists always ask themselves if proteins could be infectious or not, and prions are a perfect example. They are proteinaceous infectious particles.
I know that pro-in is not prion, but they jumbled up the O and the I to make prion. And prions have no genetic material. That means no RNA or DNA at all. They are only made of proteins. So a normal protein is generally in the shape of an alpha helix. Well, a prion protein, which we'll call PRP, prion protein, tends to be in a beta sheet conformation. So to be completely honest, we don't really know that much about prions. But it's thought that because these two proteins, the prion protein and the normal protein, are made of the same amino acids, they are the same protein, but in a different shape. When the beta sheet comes in contact with the alpha helix, it will change the alpha helix to a beta sheet. And as more and more of these alpha helixes become beta sheets, this creates protein deposits, and which is already a bad thing. But if it happens somewhere like the brain, normal cleanup still happens. So these protein deposits will be cleaned up, and that would actually leave huge holes in your brain as the proteins are removed, causing disease. And again, prions are different from viruses and viroids because, as we know them now, they are only proteins and do not have any genetic information. We know them now, they are only proteins and do not have any genetic information. Prions are different from viruses and viroids because, as we know them now, they are only proteins and do not have any genetic information.
The quality is shit, but we're gonna put it here regardless. Still about the disease, Creutzfeldt-Jakob disease. Let's see what it is. It was actually a comment down here. I've never heard of protein aggregates being cleaned up and then causing holes in the brain, which further leads to disease. This is not very clear. Would you care to explain this more? Like in Creutzfeldt-Jakob disease from the National NIH, what causes Creutzfeldt-Jakob disease? Some researchers believe in unusual slow virus or another organism causes CJD. However, they have never been able to isolate the virus or other organism in people with disease. Furthermore, the agent that causes CJD has several characteristics that are unusual for known organisms such as viruses and bacteria. It is difficult to kill. It does not appear to contain any genetic information in the form of nucleic acids, DNA or RNA. And it usually has a long incubation period before symptoms appear. In some cases, the incubation period may be as long as 50 years. The leading scientific theory this time maintains that CJD and the other TSCs are caused by a type of protein called a prion. Prion proteins occur in both a normal form, which is a harmless protein found in the body's cells, and in an infectious form, which causes disease. The harmless and infectious forms of prion proteins have the same sequence of amino acids, the building blocks of proteins, but the infectious form of protein takes a different folded shape than the normal protein. Sporadic CJD may develop because some of person's normal prions spontaneous change into the infectious form of the protein and then alter the prions in other cells in a chain reaction. Once they appear, abnormal prion proteins aggregate or clump together. Investigators think that these protein aggregates may lead to the neuron loss and other brain damages. However, they do not exactly know how this damage occurs. Copy, paste. Give me a TLDR for this. Nice. TLDR. Okay. So alpha to beta platelet sheets, they trigger misfolding. Prions are proteinogenic infectious particles. These are subviral particles, smaller than viruses. Lysosomes are the demolition and recycling center. So let's bring this here. What could be a good analogy for lysosomes in the cell from real life? City recycling centers. And recycled, broken down. Similar lysosomes in the cell act as recycling centers by breaking down and recycling cellular waste, old organelles, or foreign materials. Analogy. So, how does Golgi make lysosome? From ER, these molecules are modified sort of within the Golgi. Creation of primary. Certain proteins targeted for lysosomes receive specific mannose 6-phosphate. These type proteins are packaged into vesicles budding off from Golgi. These vesicles are known as primary lysosomes. Primary lysosome fused with other vesicles. Yeah. Once formed, so primary lysosome fused with other vesicles containing digestive enzymes, which are synthesized in the ER and modified in Golgi. This fusion process activates the primary lysosomes, transforming them into functional lysosomes. Lysosomes contain various hydrolytic enzymes that become active in the acidic environment within these organelles. These enzymes aid in breaking down materials engulfed by the cell or materials from within the cell itself. Do they work in phagocytes or macrophages? Yes, lysosomes play a crucial role in phagocytes, including macrophages. Phagocytes are a type of immune cell responsible for engulfing and digesting foreign particles, pathogens, and cellular debris. Macrophages is a specific type of phagocyte. So macrophages are a subtype of phagocytes. Phagocytes are generic, like an umbrella term for all of them. Perform this task extensively. Crucial for macrophages and phagocytes in general. Okay, we are good with this. A plasmid is if we ask that question, we don't want it again. Yeah, we didn't ask it, so we're good. A plasmid is a what DNA molecule is separate from? So it's a circular DNA molecule separate from chromosomal. So it's a small circular prokaryotic circular DNA molecule. It's DNA also. We need to make sure, and it gives advantages like superpower genetic advantages to the virus. I think bacteria. Plasmids are in prokaryotes. So bacteria are prokaryotes. Viruses are acellular. You need to know these differences. And it's a DNA. What helps the cell resist compression forces? It's microtubules. It's 13 alpha and beta dimeric tubules. Tubulins. Tubulin is the protein. What is when bacteria gets genetic info from the environment? So from the environment, fresh DNA, environmental DNA is transfusion, transformation. R strain is safe and S strain is deadly.
Welcome back.
Let's see Griffith experiments. I wish there was a I can put this here as well. So R strain, safe, the mouse survives. The S strain, you inject the mouse dies. Recovered strain is S strain. And then S strain is heat killed, you injected, the mouse survives. S strain, heat killed, the mouse should survive. And then R strain, but the mouse dies because it got the S strain, he killed, the S strain is recovered, so the mouse got it from the environment. And this is transformation. The R strain bacteria get transformed by the transforming principle or genetic material of heat killed S strain. Interesting. What is the transfer of genetic material using a bacteriophage? So, bacteriophage is with transduction, I believe, or so we have conjugation, which is not the case here. It's like a sexual thing. Then we have bacteriophage is transduction. We have transposon, which is an insertion removal of the genetic DNA. It's a DNA segment, and we also have, so it should be transduction. Yes. So transduction is the transfer gene to the bacteriophage and brings it here. Transduction. It has a lytic phage and a lysogenic phage. So a lytic phage will result in the death of the cell, bacterial death, and the lysogenic phage will make the bacteria stronger, or give it the ability to produce exotoxins, so like antibiotic resistance and stuff like that, right? What are DNA sequences that can change their position within a genome? These are transposons. Cool. Wait, there is a thing for it? Is it the same? Yes. Cool. I think it starts with transposons, right?
The bacteria has its DNA in plasmid DNA. And the bacterial plasmid is a small part of the bacterial DNA that has left the chromosome and now is floating inside the bacteria. So transposition refers to the process of extracting a small part of the bacterial chromosome to become plasmid, or reintegrating the plasmid into the bacterial chromosome.
This transposon is so are transposons related to transposition? Yes, transposons and bacteria are directly related. Transposons, sometimes referred to as jumping genes, are shaped so you can from one location in the genome to another. This ability to change positions in the genome is known as transposition. And the transposon plays a significant role in genetic diversity and adaptation. When a transposon moves from one location to another within the bacterial genome, it can sometimes disrupt genes or introduce new genetic material, leading to genetic variation. Transposition can have various effects on bacteria. It can activate or inactivate genes, alter gene regulation, or even transfer genes between bacteria, potentially contribute to antibiotic resistance or other adaptations. So transposons are the mobile genetic elements.
Keep in mind that if the bacteria want to share its DNA with another bacteria, it can only share its plasmid, not the chromosomes.
So let's take a screenshot of this.
So the plasmid is smaller and readily accessible.
So moving part of bacterial DNA from chromosome to plasmid and vice versa. To plasmid, or moving the plasmid to the chromosome. Only plasmid can be shared with another bacteria like vanillin gene.
So the plasmid is smaller and readily accessible. Just remember the transposition.
So we have plasmid for transposition. Let's put hard for this. So it goes from bacterial DNA to plasmid. So viroids are plant pathogens. Viroids cause over 20 different plant diseases, of which the most cited are potato spindle, tubular disease, chrysanthemum, hepatitis D. Viroid is a circular piece of RNA which can affect humans. What are bacterioviruses that use a tail sheath to inject DNA or RNA, and they are bacteriophages? They use a tail sheath to inject DNA or RNA. What is the protein shell of a virus? It's capsid. What are individual virus particles that are found extracellular? subviral particles. Virions, they are found extracellular. So virions, one complete infectious viral particle is a unit, like it's legit unit, contains nucleic acid, DNA or RNA. Both can be double or single stranded. Protein code, classification based on type of capsid, capsomeres, protein subunits. So virions are one complete infectious viral particle. Khan Academy link. Let's see if it's different. No, it's the same thing. So I believe we have once you put it in the clipboard, it doesn't go here. No worries. What genomes may be made of DNA or RNA and may be single or double stranded? These are circular viral genomes. They can be DNA or RNA, they might be single or double stranded. So single stranded linear DNA, single stranded circular, double stranded linear DNA, double stranded circular DNA. We have double stranded with regions of single strandedness. Hepatitis B. We have RNA, double stranded linear RNA retroviruses. This is a comprehensive one. Single strand, so it could be single stranded or double stranded DNA or RNA. We have like 2 factors, which makes 4 possibilities. And we have 5 possibilities here because there is a double stranded type, and they're all and also linear or circular. Definitely mnemonic for viral genomes. I've lost track. Here we are. Mnemonic for viral genomes. So retroviruses are double stranded linear RNA. Interesting. But here we have single stranded RNA. Oh, single stranded linear RNA. Retroviruses. Is this is there such thing as reoviruses? Yes, reoviruses do exist. They are a Reoviridae, Reo respiratory enteric orphan viruses, which reflects their initial discovery in respiratory and gastrointestinal infections and their ability to be classified initially within known viral groups. Reoviruses are double-stranded RNA viruses and are widespread in nature, infecting various animals. They can range illnesses, mild respiratory and gastrointestinal infection to more severe. This is in certain cases. One well-known member of the reovirus is the rotavirus, which is a common cause of gastroenteritis in young children worldwide. Reoviruses have also been studied for the potential in oncolytic virotherapy, where they are modified to selectively infect and kill cancer cells. Jeez. Managing infections caused by these viruses and exploring their potential application in medicine research. Who is doing research on these? Interesting. Retroviruses are single-stranded RNAs. Reverse transcriptase is needed to make DNA. HIV, positive strand, reverse transcriptase, cDNA, double-stranded DNA, integration, host DNA, provirus, mRNA or new virus, packaging into virus, budding. Very interesting. Our time is over for this, so we need to continue chapter 1. So we also had continue B1 and we also did this is a hard one, and there is a thing on retroviruses, so this is not done yet. We can do this again, but let's take a look at our deck. Let's rename this B1 cells, bacteria, virions, we had the chemistry for the IUPAC stuff. Wait. What happened to... It was here, right? Yeah. Anyway, we have 10 minutes, and then we'll get started for PsychSoc. Ciao for now.